Ladies and gentlemen, we will now move on to the last presentation of this international seminar on marine fungi. The last speaker is our good friend, Dr. Samir Damare. He is another guru of Deepsea fungi. You heard it from Dr. Welles. She is one of the gurus of Deepsea fungi. There are very few gurus of Deepsea fungi. So you heard from Dr. Patricia Welles. Now it's time to listen to another guru, Dr. Samir Damare. Dr. Samir, can you hear us?
Yes, very clear.
Oh, great. Before I hand over this platform to Dr. Samir Damre, Dr. Rohit Sharma, can you please kindly introduce Dr. Samir Damre to today's audience?
Thank you, Dr. Samir, first of all, for waiting to the last, and we are going into the last lap of our today's seminar. So Dr. Samir is a principal scientist at CSIR National Institute of Oceanography, specializing in marine microbiology and biotechnology. He has extensive experience in studying extremophiles, particularly those from deep sea environments. His research includes bioprospecting for novel bioactive compounds and understanding microbial ecology. Dr. Damre is skilled in various molecular biology techniques, contributing significantly to the field of marine biotechnology. His work is crucial for discovering new microbial resources and their applications. Thank you, Dr. Damre, for joining. Please go ahead with your talk.
Yeah, the slides are visible and I'm audible to all.
Yes, Dr. Damre, both are fine. Yeah.
Okay. Thank you, Rohit and Belle, for giving me this chance. And thank you all the people for hanging on to the last talk. So in continuation with what Patricia spoke some time back, I'll be giving a little more detail about the diversity, culturing and applications of deep sea fungi. Before I go on to my talk, that's my institute where I work, National Institute of Oceanography. This is the building where I'm sitting at present. So we have a headquarters in Goa, and then we have regional centers in Bombay, Cochin, and Vishakhapatnam, where Dr. Belle works. We are really lucky to have two vessels with us. Our institute owns this vessel. This is a coastal research vessel called Sindhu Sankalp, and this is the ocean-going research vessel called RV Sindhu Sadhana. So most of the deep sea work what I have done is carried out on this vessel. And during my PhD time, when I was working here in NIO on some foreign chartered vessels. So we need at least like 40-45 days of endurance to go in any of the deep sea expeditions. Yeah, we have all been hearing this since the school time, that oceans cover more than 70%. And what I'll be talking today is more about the Indian Ocean, that's covering 20% of all the oceans. When we talk about the marine environment, based on the light zones, we can divide them into the coastal zone, the tidal zone, the difference between the high tide and low tide. Then you have the coastal zone. Then you have the continental shelf. And this is the euphotic zone, the first 200 meters, more or less, including the deeper parts of the ocean, where you have all the primary productivity going on. So this is where your CO2 fixation is occurring, and the phytoplankton are converting it into the food, which ultimately is being eaten by other organisms and the food chain derives. Then you have the bathyal zone, where we get a lot of the fish what we eat most of the time, like this kingfish and pomfret and prawns and many other fishes. And then below that, you have the abyssal zone or the dark zone, where there is absolutely no light. And this is what is unexplored region. I mean, you have a lot of space explorations going on, but there are not really many missions which are going down to explore this abyssal zone. And that the deepest point, as you know, is Mariana Trench, described one. They might be deeper than that also, we never know. So this is the place where all the life which is surviving is close to mystery, you can call, because we really don't know how they survive and what are the things occurring there. So talking about marine fungi, we can call them as obligate marine fungi or facultative. So obligate are the ones which complete their complete life cycle in the marine conditions. So when I say marine condition, basically you can call it as a requirement of the salt as such, or the facultative ones are the ones which can grow and survive in both the conditions, in the marine as well as devoid of any salt. We won't go into the further descriptions about the temperature and pressure, then it becomes very complicated. So this was described by Colmer and Colmer sometime in the early 60s. In a previous talk, Patricia spoke about this. So only the year was missing. So 1964 was the first marine fungi which was isolated from 4,450 meters. So it's almost close to 60 years, but in spite of that, we really don't know much about it. It's a totally unexplored. Then in 1979, that is where the definition of this marine fungi came into. Obligate marine fungus was isolated by Colmer from some of the wooden panels which are immersed in seawater, and they could close down the complete life cycle in the marine conditions. And these cultures, what they studied, were not able to grow without the salt. So that's why they called them as obligate marine fungi. Then came the second phase of the high pressure cultivation of this marine fungi. So 1992 was a time when a couple of publications came out. Lorenz and Molitoris was the first one to publish. And then my PhD supervisor, they reported in Deepsea Research Journal and fungal isolates from 965 meters from Bay of Bengal, which are isolated from some calcareous shells. And 1998, a detailed study was carried out with regards to their life cycle with the same culture which was isolated from this Bay of Bengal. And then in 2006, we came out with a paper describing the first detailed diversity of this culturable fungi from the deep sea sediments. So when we talk about the deep sea, this is one of the extreme conditions which could be like something similar to the deserts, polars, or the polar oceans or the volcanoes. So basically, these are the environments which don't support the life so easily. So what is so difficult for survival in deep sea? First of all, as I said in my previous slides, the constant darkness. So there's absolutely no light there, because of which there is no productivity occurring as such. So the food supply is always short. So that is the sporadic occurrence of nutrients. We sometimes call this as a feast and famine condition. So like in deeper parts where you have absolutely no food supply going on, suppose if there is a death of a big fish like a whale or a shark or something, the degradation rates are very slow because the organisms are also less in number. So that is the time when we say that, okay, it's a feast condition. And when this food source gets over, the organisms again need to adapt their metabolic activities to the famine condition. Then you have high hydrostatic pressure. I'll talk about this in the next slides. And you have the temperatures ranging. I mean, once you cross around 2000 meters, you have a uniform temperature of 4 degrees in the deeper waters. So it's like a huge refrigerator where all the life comes. Like many of the times you see that if you want to preserve a culture, you put them at a low metabolic activities, you keep them in the refrigerator. So that is what is happening in the deep sea also. You have these cultures which are stored at 4 degrees Celsius, but in spite of that, many of them are showing activity and growth in these conditions. I said about the pressure, high hydrostatic pressure. So this is the example of range of pressures which are present in various biological systems. So this is one bar where we all are sitting at present. I'm presenting you, people are listening to me. So that's the atmospheric pressure. You have a negative pressure in the xylem of the trees. That's the reason you have the water which goes from bottom, from the soil up to the tip of the leaves. So that occurs because of the negative pressure. Then you have fish swim bladders, the fishes which are surviving in the deeper parts of the oceans. And then you have very deep places like say Mariana Trench or in Pacific, where you have very high pressure. So if you correlate it, it's like every 10 meters of the depth, you have an increase of 1 meter. So when I talk about, say, I isolated organism of 6,000 meters, that corresponds to around 600 bar of pressure. That's 600 times of the pressure what we are bearing at present. And in such high pressure also, the cell wall and the cell membrane remain intact and the organisms are existing. If we are put into such pressures, we will collapse. In most of you must have read about the Titanic accident which occurred a few months back. That was because of the pressure, they say. So coming to the questions which we asked ourselves, like, are fungi present in deep sea sediments? If they are present, do they grow and survive in these conditions? And if they are growing and surviving, what is the potential possible role they might be playing in these deep sea sediments? So coming to the first question, whether they are present in deep sea sediments. Now, to see whether they are present or not present, first and foremost thing is we need to collect the samples from the deeper parts. So here we will restrict ourselves. I'll talk only about the water and sediment samples which we are using for culturing of this fungal isolates. So for many of you who do not have background about the deep sea and the marine, I'll spend some time about how the samples are collected from these deeper parts. So this is called as a Niskin sampler, what you see here on the right side of the slide. So this is the basic simple bottle which is open from both ends, and this is attached to this attachment which is called as a rosette sampler, and which is having some instrumentation which records the conductivity, temperature, depth. That is what we call as CTD. And this is sent, if you see minutely, you can see these ends are open, and this is sent down with the cables. So whenever I say that, okay, I work with deep sea samples, the first question people ask me is that, do you dive? No, that's really not possible. I think the deepest dive which humans have done without any support or anything is 100 meters, and even our nuclear submarines don't go to that depth. You need a special underwater vehicle which can go to such places. So it's most of the time we are on the ship and we send all the samplers down to collect the sample for us. And when it reaches the desired depth, we have trigger on the deck which we press, and then these two ends close and the water from that particular depth gets entrapped inside. So you get that water without any contamination. This is leak proof, so you don't have any ingress of water from the surrounding environment into it once the bottle is closed. And then this bottle is bought on ship, and then we do all our further microbiological culturing and other studies. So this is the simpler sampler named after the scientist Niskin who had described it. Then the next is we have for the sediment. That's the, you can say, the soil which is there in the deeper parts at the seabed. So there are many samplers available for that, unlike this Niskin sampler, which is the most used even now after so many years. There are not many modifications done to it. So this is called as a Usnel box scorer. So you actually have this orange thing, what you see is open, and when it hits the bottom of the sea surface, this closes, and when it closes, it scoops the sediment from the surface into the box inside. Typically this box is of a size of 50 centimeter by 50 centimeter by 50 centimeter. So you get a bulk of sample, and this box is bought up. This have drawback, like when it is coming up, there's a possibility of contamination of the water going in, but then we do take care of that. I'll explain you in the next slides. Then this is something called as a multiple corer, which you will be using these days. So you don't have the problem of the water coming in this samplers. So you are pretty sure that whatever cultures you isolate from the sediments which have been collected in these tubes are from the deeper parts, and they're not from the water which has entered into it from its journey to bottom to top. So this is at present being used by most of the researchers who are working with the culturing or the deep sea biogeochemical sampling. But then here you can either here or here, you can just collect the top 50 centimeters sediment, which is the most biologically active sediment portion. And as you go deeper down, you have less activity because it starts getting compact and there is absolutely no very less nutrients. But then if you are interested to study that, there are different equipments which are available for that. This is called as a gravity corer. So you have this huge weight which is attached to it, and this is a long tube of around 6 meters. On an average, 4 to 6 meter tubes are being used for a gravity corer. But then wherever you are planning to use this, you need to have very soft sediment, because what happens is with the weight attached, like this is close to 2 tons of weight which is attached, 2000 kg. This goes down, and because of the wet, this gets pushed into the sediment, and this is how you can see. This is the pipe which is got out and then cut in the laboratories. If you still are interested in more deeper samples, then I know most of you must have seen this borewell drilling. So this is something very similar to that, where you drill, and here we can get up to 20 to 25 centimeters on our ship, of course. But then there is a program called as International Ocean Drilling Program, where they have got cores up to around 1,800 meters also. That's like 1.8 kilometers long core, and the target of the expedition is to just go in one place and drill like 2 to 3 months to get very long core, which gives a lot of information about the climate change and the life existing and the origin of the life, etc. So it's a IODP program which is run by the United Nations, where many people can apply and they can be a part of it. And then you heard in previously Pratisha's presentation, they use something called as autonomous vehicle, where you have arms and that can collect. So this is not available with many people. Jamstick in Japan, French and US they have. We in India, we do not have, but if you are following the news and the recent Twitter, you will see that the government of India has launched the mission for building a autonomous underwater vehicle, which is capable of going to around 6,000 meters. So something similar to moon mission, they have launched the ocean mission, wherein they plan to do the sampling, and this is being indigenously built at present in National Institute of Ocean Technology in Chennai, which is which comes under the Ministry of Earth Sciences. So most of my work, what I am presenting here, is being done using this spade corer. This is the box which comes up, and then this is the kind of thing. That's why I said like the center part here is not very contaminated, and after that we take it to the laboratory, cut it into different sections. That is more important because in this area where we have done sampling in the deeper Central Indian Ocean, it's approximately takes like 1,000 years for 1 centimeter of the sediment to aggregate. So if you say something like 30 centimeters you are doing sampling, it's straight away close to 30,000 years of old organisms we could be finding. And except for the top 5 centimeters, you don't get mixing of the sediment so much. So it remains really intact as it would have been there 30,000 years back. After we get to the lab, we cut it into the sections. Then we go for the culturing and different methods of isolation we have used. Obviously, the traditional dilution plating. Then we have done something called as a particle plating. Most of the people who work with mangrove fungi and mangrove sediments, they will be aware about the particle plating technique, where particles in the range of 100 micron and 200 micron are plated, and the fungi associated with those are given a chance to grow. And then we have developed in our laboratory during my doctoral work this modified enrichment technique by incubating under the elevated hydrostatic pressure and low temperature. I'll talk about it when I talk about the results of this. So using these different techniques, we were able to isolate close to like these are the different samples which used, and if you see the recovery, like from 416 samples we used, we could recover around 94 of the sections showed presence of fungi. It's close to 23, where the dilution plating and pressure incubation, we got very good recovery. And we were lucky to get some organisms also in the samplers, in our sediment sampler, as well as the multiplankton net. Unfortunately, I forgot to put the picture of that. But multiplankton net is the one which is used for collecting the zooplankton and phytoplankton, zooplankton mainly from the deeper parts. So those organisms were again plated on the media, and we could see a lot of fungi growing on them. So this is published. The results of these are published in 2006 in Deepsea Research. This was one of the first papers describing the culturing of the fungal deep sea sediments and the diversity. So most of the samples what we collected in this studies were from 5,500 to 6,000 meters. These are some of the fungi which have been reported from the deep sea sources. in our work, we could get most of that. Just the previous question you heard about Dr. Belle asking Patricia about the Aspergilla and Penicillium. What happens is, like, most of the time these are present in the form of spores in the sediments. They get deposited. I have made a cartoon of that. You'll see in the next slides. And when they get the right conditions, they start growing. That's the reason most of the time you end up, and initially when you grow them in the laboratory, they don't look like a typical Aspergillus or a Penicillium. As you keep on subculturing, this is what we call them as training to grow in the laboratory conditions. They start behaving and coming to their original form of Aspergillus. Many times you end up thinking it's a contamination, but that is not the case. It's actually Aspergillus, which grows in different forms when it just comes out of the pressure. So these are the different cultures which we could get from the different sections. Representatively, we have put only up to 40 centimeters. Some of the sections we got up to 50 centimeters also. If you see, most of them are Aspergillus, Penicillium. There are a lot of non-sporulating fungi, which are then a huge amount of unidentified sporulating fungi, because most of the time you have your keys which are made to identify the terrestrial fungi. So the keys many times don't work with this, and it's difficult to isolate. But now with the present improvements in the molecular biology techniques and the NGS, it's becoming easier. But this is the old one which we had worked upon. Apart from doing this culturing, we also did a direct observation of this sediments for the presence of fungi, and we could get good number of mycelia and the spores germinating in this. This is a technique which we developed in laboratory to observe the fungi. If you see here, this is fluorescing little bit in blue in color, and then you have all this muck surrounding it. So if this was not fluorescing, we could have mistaken it for some dirt. But then there is a simple way to, this could be adaptable to your fungi from the mangroves or any other sediments also. You can see the details in our paper, but in short, what we do is we get this sediment and then stain it with a optical brightener called as chalcophore. So this basically stains the chitin in the cell wall, and then we observe under UV. This is the fluorescence what we see, and it becomes very, very easy to observe this fungi and identify them and confirm that, okay, these are the fungal mycelia, fungal hyphae, and not some dirt material. There is a drawback, obviously, because if you are using it in the mangrove conditions, you will have a lot of leaf litter and the plant material also, which will be having chitin. But in deeper parts of the sediments, deep sea sediments, we do not have that interference. So it was very much easier for us to see and count the number of fungi or the biovolume of fungi and the length, breadth, which we use to calculate basically the carbon input of this fungal mycelia in the deeper sediments. So you can try out this method if you have less of the leaf contamination issues. When we sent our paper in 2006, ultimately it got accepted. But first, when we communicated a paper somewhere in 2003, the major question which was raised by all the reviewers was how sure you are that this fungi are from the sediment. And as I was telling you, the drawback for the sample, not from the water, because ultimately it travels through 5 kilometers of water when it comes from the deep sea bed up to the seawater surface. And there's a possibility, it's not wrong, that okay, there's a water contamination and the fungal spores or hyphae which are floating in the water, they enter into a sampler and they contaminate the sediment. So what we did is that we took the most commonly isolated fungus. Fortunately for us, it was not very difficult to grow. It was Aspergillus terreus. From the name it says terreus, but yeah, that was the one of the most abundant fungi which we found in our sampling. We have done like around 7 to 8 expeditions in this area, and most of the time we get Aspergillus terreus as the most dominant ones. And we made some antibodies against these cultures, and we went back to the original sediment section from which this culture was isolated. We took good care about doing a lot of cross immunity studies to see that there is no cross reaction to other fungi, and we were lucky that the method worked out. And as you can see here, these are the original filaments which we could see from the sediments. and we found some organisms also on which these cultures are growing. And after this was done, that is the time, after 2.5 years of struggle, we could publish the paper, and the reviewers accepted that whatever we are reporting is from the deep sea sediment and not from the contaminant water column, as not as the people believe that okay, it contaminates. We did some more experiments of that to see whether they're because antigen and antibody work with the structure of the lock and key mechanism. So what we did, we just did culturing of this the spores what we collected from Aspergillus terreus and tried growing them on the in the atmospheric condition, and we could see that the reaction was still intact. So maybe it is not affecting the binding sites from against which they are produced. So if that was there, we wouldn't have got this reaction. So after all these studies, we postulated this hypothesis where we say that, okay, how the fungi reach the bottom of the sediments. Like these wind-blown fungal spores or mycelia, they come, get deposited on the ocean surface. They might aggregate with the marine snow or other any thing, and they might be taking months or years to reach down into the sediments. And after that, as I said, like the sedimentation rates in most of the deeper parts of the oceans are not very high, as you see in the coastal areas. Like the area where we are doing sampling, as I said, 1 centimeter takes around 1,000 years to get accumulated. And this slowness of their travel and then the staying in those conditions, they get adapted to these conditions, and like low temperature, high pressure, and low nutrients. And in spite of that, some of them still remain dormant and some die. So the ones which remain dormant could be the ones which we recover when we give them right nutrient conditions. I say in terms of the food and the temperatures from which they have originated. And this theory was well supported by one publication which came out from the Caribbean sea fan extinction, which occurred somewhere in late 90s. And the researchers traced back the origin of the fungus which came, Aspergillus it was, to the Saharan desert. So these fungal spores and mycelia actually can travel those distances, and this could be the reason why most of the time we end up getting the most dominant forms, because Aspergilli are very sturdy and they survive these extreme conditions and the spores of them. So with that was enough to say that okay, the fungi are really present in the deep sea. The second question then we had was, do these fungi grow in the deep sea sediments if they are present? I mean, they could be present in the form of spores or the mycelia, but do they have really that much of metabolic capabilities to grow in this extreme conditions? Just a minute. Yeah, but unfortunately, there is no report of a true piezophile. That's the fungus which can grow only at high pressure. There are some bacterial cultures which have been described and they grow only at high hydrostatic pressure. And when they are tried to grow at the room temperature, room pressure, atmospheric pressure, they do not survive and grow. So most of the fungi what have been reported are piezo tolerant, means the pressure tolerant. Like they might be growing or not growing in the high pressure, but they are capable of growing at the atmospheric pressure. So these are called as a piezo tolerant or barotolerant. Like barotolerant is an old word. People use piezo these days. Piezo is for the pressure. This is what we have in our laboratory, which is called as a high pressure microbial culture vessel. So this cylinder, what you see here, we put our cultures and then close it. And this pump, it gives the hydrostatic pressure whichever we want for. So many of the cultures what we isolated were capable of growing at around 600 bar, obviously because they were isolated from that depth of 6,000 meters. Unfortunately, not many cultures survive the subculturing in the lab. So they could be the true piezophiles which are there, but we do not have mechanisms to sustain them for longer time in this, because this is an enclosed thing for which you have nutrient limitation, oxygen limitation, Many people ask me that, okay, when you are going in deep sea, there is enough oxygen for the culture? Yes, the waters are saturated with oxygen. We have reported close to 7 to 8 ppm of oxygen in the deeper waters also. So it's not that they're devoid of oxygen. So they do need good condition, but then the metabolic rates are so low that the oxygen which is present in this is enough for them to survive for months together. So most of the time when we come back from the cruise, the first isolation incubation is always close to 30 to 90 days, because we need to give them time at their metabolic rates, what they are used to, and then we start to train them to grow in the laboratory slowly. So like for the spores which we get from these deep sea fungal cultures, or the spores directly from the sediments, we could see that the germination spores reduces with the increasing hydrostatic pressure. Obviously, this is for the spores which are produced at the atmospheric pressure, because in the submerged conditions, they don't produce the aerial spores as such. We could not see germination of spores at low temperature and atmospheric pressure and elevated pressures. The viability reduced by around only 3 to 5% of spores, but they could remain viable at low temperature, after which it was really constant. So these are the spores which would be surviving in the deeper conditions. We did monitor this for around 1 year, and we could see that after 1 year also these spores are viable when given the right conditions. So this is what must be happening exactly in the deep sea conditions. So it is like basically the low temperature which is more inhibitory to the spore germination and growth than the elevated pressure, because the organisms are quite capable of growing under high pressure, but the low temperatures are the ones which are reducing the metabolic activities. We did some experiments with regards to the biomass production, and we could see that cultures produced at almost equal biomass under different growth conditions when the inoculum was in the form of growing mycelia. What we did was we grew the mycelia in the atmospheric pressure, put it back in the high hydrostatic pressure, and then we could see that there was not much difference. But when we used the spores as a starter for the culture, we could see that not many spores are able to germinate in the low conditions. That is where we started this idea of using some stimulants to grow, because we thought that, okay, the nutrient concentrations could be the one which are affecting the spore germinations. DMSA is one of the known stimulants for the germination of spores, but here in these deep sea cultures, we saw that we couldn't get any increase in the germination with the use of DMSO. One thing which we saw, that when we gave a heat shock to the spores for at around 50 degrees for 15 minutes, the germination percentage increased drastically, but then above 60, it was quite lethal, and nothing grew above 60 degrees. We also tried some stimulants which are normally used for the terrestrial cultures, like sugars, glucose and sucrose. We could see that they helped improve the spore germination rates quite drastically. Another thing which we noted in our studies is that, like, many a times when we go for doing some isolations, we just come back to the lab and use the normal microbiology media which are being used, like say it could be a Savarots dextrose, potato dextrose agar, malt extract or anything. But in our study, we found that many a times this high concentration, high nutrient media, what we use for isolations, are actually toxic to these cultures, because in the deep sea, you have very low nutrients and it becomes a nutrient shock to them actually when you feed them with so much of food. Because most of the time these enzymes are the low km value enzymes which have very good efficiency and get saturated very fast. So what we did is we collected the sediment from this region and we did some extractions from this using different extractants. And this one we filter sterilized to remove any other organisms. And then we could see that the growth of the fungal isolates from this region was much better or similar to that what we use using the laboratory media. But here also the lab media what we used was 1:10, 1:50 or 1:100 diluted. We avoid using direct concentrated media when we are doing isolations from such low nutrient at the environments. So with this, we have successfully answered the first two questions about the fungal presence in the deep sea and their growth. And if they are growing, the third question comes is, how do these fungi survive in these deep sea sediments? What are the survival mechanisms? So survival mechanisms and adaptations could be divided into two parts, basically morphological adaptations and then you have physiological adaptations. Under physiological adaptations, you have many adaptations which have been reported from the deeper organisms like membrane composition, organic osmolalite production like trihalose, glycine, betaine, ectoin, sucrose, mannitol, glycerol, TMO, etc. And then there is stress protein, which are like universal stress adapters like heat shock proteins and cold shock proteins. The examples what I have given her have been reported many a times from the higher organisms like fishes, which have been found in the deep sea conditions, whereas the other ones, membrane composition, stress proteins, have been reported from some of the yeast and the bacterial cultures. This is what I was talking about, the first isolated pictures what we get. This is what we have been taught in mycology, like this is how aspergillus looks like. You have phyllites, you have conidiospores, you have conidiophore, and then conidiospores come into that. But these are some of my pictures which we got after the first opening of the pressure vessels. Under the microscope, you said something? Well, so you can see that you have these typical structures and you have a conidiophore and then again another conidiophore coming out of it. Obviously, because they're submerged in water, you don't have these aerial conidiospores as such. But these are the different structures. And then after you grow them in the laboratory for say 3 to 4 subcultures, you start getting a normal growing aspergilli. And that is why I said like you need to train them to grow in the laboratory, do some experimentations with it. These are the different adaptations which we could see when these cultures are grown under pressure. Like this is a normal one where you could see normal hyphae, and then and when you start putting the stress onto them, like this is an example of 200 bar of pressure or 20 megapascals, and 5 degrees 1 bar and 5 degrees 200 bar. So as I was talking in my previous slides also, it's mostly the temperature we found is affecting it more than the pressure. From these pictures, photomicrographs we could see. This was one of the deep sea yeast which we got and which was growing quite well at even 600 bar. Unfortunately, after 6 or 7 subcultures, it didn't survive in the deep sea conditions. Obviously, it could be the nutrients. We really don't know how the nutrient cycle works there, because if you talk to any of the biochemistry people about the nutrients, they say that okay, nutrients affect very badly. Like the normal, say, yeast extract or malt extract what we use, they get very badly affected with the pressure what we are putting on them to grow these cultures. And that could be reasons the organisms or the enzymes which are produced by these organisms are not able to utilize these nutrients what we are giving, because obviously the lock and key mechanism of again the enzyme substrate may not be really functional there, but not much work has been done on this. So these are some of the extension. This is very normal. Like when you put a stress on the yeast, you get these elongations and also it's not only pressure. If you try any other stress also, you might get this. But these are some of the observations we got. We could also see some microconidiations occurring. It's similar to the previous slides where I showed you those aspergilli pictures. So you have a spore, then you have a growth, then you have again sporulation, again growth. This is how they try to overcome the conditions and propagate themselves under the high hydrostatic pressures. This is universal protein stabilizers. It's called trimethylene N oxide, which has been reported from some of the mussels of Tiloes, cats, and crustaceans. It has not yet been reported from any fungus so far. Another aspect is the membrane fluidity. All of you know the saturated and unsaturated ratio, like coconut oil gets frozen. So something similar happens to the yeast membranes also. That's the reason they keep on changing the ratios of the unsaturated fatty acids and saturated fatty acids within the membrane phospholipids, so that the membranes remain in more fluid conditions even at the low temperature and high pressures. This has been successfully shown in many of the Saccharomyces cerevisiae experiments, like how we have E. coli in the bacteria, Saccharomyces cerevisiae is the workhorse for all these kind of experimentations with regards to the stress or other conditions. Trihalose is the one which has been demonstrated successfully, which protects the organism from dryness, freezing, pressure. It stabilizes the membrane and other assemblies under the extreme environmental conditions. But then when we did experiments with that culture, which I showed was able to grow at 600 bar, we could see that the trihalose production was not much seen. So maybe that particular culture had some other mechanism to grow. And that is where we started looking at the next aspect, that is the pressure responsive gene expression. We couldn't do much, we couldn't get much head-on start in this. So we went to the next point, that's about the expression of proteins. We did some experimentations, and yeah, we have a nanochip LC MSMS QTOF in a laboratory where we have grown the cultures in different hydrostatic pressures and then studied the protein profiles. And we could see that most of the proteins were seen in cultures grown at once compared to the elevated hydrostatic pressure. Proteins detected at 5 degrees Celsius of pressure, the stress were much more than at 30 degrees Celsius. But again, the proteins produced at 1 bar were more than 100 and 500 bar. This could be the reason, because the metabolic activities are 1 bar is quite high as compared to 100 and 500 bar. But we could see a substantial difference in the proteins which are being produced by these cultures. This paper is under communication with a journal. So once it is out, you'll be able to see the more different things. But then the traditional stress proteins which people describe, we couldn't isolate any of those. So maybe some other proteins which we found as elevated expression could be responsible for these cultures to grow at the high hydrostatic pressures. So these are a couple of papers with regards to this high hydrostatic pressure, and there is one thesis my student has submitted. This is available on the website. You can have a look at it. So there you will get more details about the kinds and the numbers of proteins which were being produced when these cultures were subjected to high hydrostatic pressure and low temperature. So a lot of more work needs to be done with regards to the protein expression as well as the transcriptomics, which we haven't touched upon yet. So for the questions which we raised at the start, fungi are present in the deep sea, fungi survive. These are the papers from our group. You can see from 1998 to 2012. After that comes the protein expression studies. So coming to the last part of my talk is what is the role of this fungi? If they are present, they are growing, and these are the different mechanisms. What could be the possible role of this fungi? In the previous talk, you heard Patricia talking about their role extensively in the biogeochemical cycling and even some degradation studies and all they have carried upon. We have done some basic work with regards to carbon sequestrations. So basically carbon sequestration, nothing but it's the aggregation. This fungi could be helping in the aggregation and recycling of nutrients by means of the degradation of the complex molecules in the simple forms. Apart from that, we studied these cultures for production of novel enzymes, and one of them we have described in detail, the cold tolerant proteases, which could be used in the detergent industries. I'll give the details in the coming slides about it. So the aggregation, when we talk about, this is common for deep sea or in the terrestrial environment also. It helps in the stabilization of the sediments. It helps in the carbon sequestration, of course. Formation of humic enzyme complexes so as to facilitate the nutrient cycling in the sediments. Because when the organisms are producing the extracellular enzymes in the water environment, what happens is they go away from the organism once they are secreted. Like oceanic, the water column is so huge. So what is the point of these cultures producing these enzymes when they are not going to be any helpful to them as such? So that is where these humic enzyme complexes help, and the substrate gets entrapped in this, and that is where the enzymes can act upon them and the cultures can feed upon the degraded products more easily. And the last one is dot, which we will see in the next slides. When I was talking about the fungal presence and their detection method in the sediments, what we use with the calcrophore, So with the help of that, we could calculate the bio volume of this fungal biomass, and we could see that it contributed around 2.3 to 6.3 microgram carbon per gram of dry sediments. Now this, if you say, is much less than the bacterial contribution, but nevertheless, this is huge in terms of, like, initially fungi were not at all considered for this role. But then we can see that this fungal biomass has a bigger role to play in the carbon sequestration and the bio volume. The results of this are available in the microbial ecology paper which we published in 2008. But then why they were neglected all these years? This could be one of the potential reasons why the researchers who are working with bacteria and other sediment, like geologists and all, why couldn't they see any fungi? Like if you see on the left hand side, you see this structure here. If I tell you there are fungi in this, people will not agree to it so easily. They will say, where is the proof that? And that is where this treating of this sediments with EDTA, as you know, EDTA is a chelator. So it chelates all the metal cations, and that is where you start seeing this hyphae more. But care has to be taken that the EDTA concentration should not be very high. Otherwise, your fungal mycelia will disappear in front of you. It happened to us when we are standardizing. We are observing under the microscope, and suddenly the fungal mycelia started disappearing. And then we have standardized. All this is available in the 2008 paper, and we could see that more than double sediment subsections showed the presence of this fungal hyphae when we use this EDTA treatment. Initially, when we did observation of the fungal hyphae in that 416 sediment subsections, we could see only in 5%. But after this EDTA treatment, the percentage more than doubled up, going to 12.8%. And it could be still furthermore also, because ultimately we are using a small, like 0.5 to 0.1 gram of the sediment. So the message from this is that whenever you are seeing something like this, don't give up. There could be the presence of fungi which is lying embedded, because as you see, to prove this, we did an experimentation with the sediment extract, which was 0.22 micron filtered. So absolutely no particulate matter into it. There was only dissolved nutrients present in that. And you could see that slowly during the growth, this Aspergillus terreus, which you had got from the deep sea sediments, was started secreting some kind of exopolysaccharide, which started trapping the dissolved nutrients which are present and forming the enzyme humic complex, which is ultimately, this is how the mycelia look like, and could be helping for the nutrient degradation into this humic enzyme complexes. So from the clear liquid, we ended up with this, which conclusively proved that, okay, these are the ones which are responsible for the aggregation inside the in the water column. Also same thing might be happening, and in the sediments also. This is just an SCM image to see the difference in the EDTA treatment. These are the fungal mycelia, and then once they are devoid of all these aggregates around them. We did some experiments with regard to alkaline phosphatase activities. And as I was saying you, these are really good candidates for the discovery of cold active enzymes and byproducts, mainly because if you see the enzyme efficiency, the enzymes with low Km values are more efficient because they reach their Vmax quite fast. And this is what is happening in the deep sea conditions also. Because the nutrient concentrations are so low and the oligotrophic nutrients, the enzymes have to be really more efficient. So if you need the enzymes in the industries, low Km enzymes are preferred for higher efficiency, because then you need low substrate to get the product out. So these are really good candidates. And this is the cold tolerant protease which we described from one of the fungus. We tried using it as a detergent additive, and we could see that it was able to remove even spinach and gold stains from the cloth in less than 15 to 30 minutes of time even at room temperature. Actually, this deep sea fungal work which I started was the origin of this was like department of the DRDO and Indian Army. They have their bases in the cold, like -20, -40, the glaciers and all, where the human waste doesn't get degraded so easily. You know that you have a septic tank where all the organisms attack, but the temperature is so low that the human waste doesn't get degraded. That is how they originated. And the second one was many of these temperate countries where you have cold waters coming in, you have to spend a lot of time and energy to heat up the water to wash your clothes properly. And normally that's what we do. If the cloth is too dirty, we go home, heat some water, and then use that hot water. But instead of that, if you have a protease or a lipase enzyme which is able to act as efficiently as at room temperature at even colder waters, you will be saving a lot on energy. That is how the research about these enzymes started in our laboratory. We have Indian, European, and Japanese patents for this culture, and a couple of industries have been using this enzyme in the detergent as a detergent additive. But then, as I said initially, you have a lot of space explorations as compared to even deep sea explorations, because the deep sea exploration is very costly affair. Like just to give an numbers, the when we go on an expedition, the expenditure for 1 day of our sailing is costing around 17 to 20 lakhs. This is excluding of the manpower and other, just the sailing cost. So you can imagine, like if you sail out for say 30 to 45 days, you are going to end up spending something like 7 to 8 crores just to collect the samples. So it's really, if you say that, okay, I'm going to study the deep sea fungus from this region, nobody is going to fund you. I was lucky to part of this environmental impact studies which Government of India is carrying out for the deep sea mining, which so most of the work was carried out under the environmental impact studies, where we wanted to establish the baseline of these organisms which are present in the deeper parts. So that is where we got a good opportunity to carry out many expeditions in this area and try out new things in the each expedition for isolation and other studies. This is just to give a close-up all the samplers, what I described in my previous slides. So this is that Liskin sampler. This is around 12 liters in capacity. This is that multiplankton net, what I spoke about, which is used for collecting the zooplankton from the deeper depths. So this goes down, and then when it is coming up, it catches the organisms get trapped. The size of this is around 100 microns in size. So anything bigger than 100 micron gets trapped here, and then we can get it on the lab and study for their presence and the associated organisms. This is the sediment. This is the sediment which we get from this. If you can see, there's a difference in the color also. Now, from the area what we have been sampling, there is a record that this whitish part is actually a volcanic explosion. This is there's a lot of ash content in that. We have got fungi growing in these parts also. So this is how it looks like when it comes, and then you take to the laboratory and cut this section. This water is the most what do you say? You can do a lot of, because this is the interface water, what they call, and all the biogeographical cycles, the reactions are occurring in this. So at present in my lab, apart from this deep sea stress work, we are working towards the creation of the proteome map for the Indian Ocean, because ocean metaproteomics is a very fast emerging field which is enabling to discover a lot of marine microbial communities and their impact on the global. So we are carrying out metaproteomics sample of complex samples such as seawater, sediments, then sinking particles, some of the biofilms which were collected, and the organisms which we have found. This is another second program. That was one environmental impact assessment. This is the second program which I was lucky to be part of, and led a long expedition of 90 days from March 14 to June 6, 2021, where we did this red line. What you see is one non-stop journey which we did, and we collected over 700 samples. So which we are exploring for bacteria, fungi, metagenome, metaproteome. So all kinds of omics studies we are doing with these samples, and another couple of years time, I think we should be able to build a huge genome and proteome database for this region, which has been neglected so far. This is my team which sailed with me under the program called Trace Biome. If you are interested, Google out Trace Biome. You will see you can see the documentary which the Doordarshan people have made for our expedition. 2 hours of English and 1 hour of Hindi. So you will have an idea that this, when we say cruise, it's not the Hollywood or Bollywood kind of cruise. A lot of work is being done on this. So this is my team which sailed with me for 90 days. So if you people are interested for joining NIO for your PhD, you can check our website for the details. And it's ideal that you clear your UGC CSR net and come. That way you can join directly. Because if you join on a project as a project assistant, you need to work for 2 years at least before you can register for a PhD. So that becomes a 7-year affair. So the easiest option is to join after you clear your net exams. I do have 2 positions open with me for PhD. So if anyone is interested, they can write to me. Or if you just want to visit NIO, you can do it through your college or university. With that, I end my talk. I would like to thank my PhD supervisor, Dr. Raghu Kumar, and her husband, Dr. Sheshagiri Raghu Kumar. As Belle was saying, me and my wife, I worked with her and she worked with him. And these are all my students who have contributed to all the data what I presented and what we have collected in last 22 years of my stay at NIO. With that, I take leave. Thank you.
Oh, I think that was a brilliant presentation, Dr. Samir. Very energetic and full of passion for Deepsea. I have a small comment. You asked 3 very interesting questions, right? For the first question, for the presence of fungi in Deepsea environment, I think you guys generated lots of evidence in support of that, in support of the statement that fungi do live, fungi are found in Deepsea environment. The second question is, do fungi grow in Deepsea environment? I don't think the evidence provided is quite feeble. What are your views on that?
We have not completed the life cycle of fungus. If you call in terms of the growth, I do agree for that. What we have done instead, we have looked into the biomass generation, like the growth, which is one of the ways to measure the fungal growth. And we could see that there was an increase in biomass production when the fungus are put under this pressure. And we do have a lot of controls which are maintained to see whether it is just like the normal growth or it is because of the stress which is there. Biomass production as compared to the atmospheric pressure was less, as I have been saying repeatedly, because the metabolic activities are quite low. But then yes, there was an increase in biomass, and this could be one of the conclusive things to say that the fungi are able to grow in the biomass. The second question was whether their spores, if they're the non-vegetative forms, are they able to germinate? And we could show in our lab experiments using the pressure simulator that the spores were also able to germinate. Unfortunately, the germination percentage was very low. That's the reason we had to use the stimulants. And some of the stimulants are not reported from the deep sea, but these we had to do to check whether they are able to grow in this and germinate, not grow, germinate at the deeper conditions, simulated deep sea conditions.
My last question from my side. And do you find only filamentous fungi in deep sea, or do you find yeast forms also?
No, we have got lot of yeast, but unfortunately, most of the yeast we are not able to sustain their subculturing. One of the yeast, what I did a lot of experiments, was able to grow even at 600 bar, but then it was not able to grow a lot in the culture, and we have lost that culture now. We don't have it anymore.
I think, but we must have enough DNA signatures for the presence of yeast forms in deep sea, right?
Yes, we have recently, the expedition, what we have done a lot of sequencing where we are seeing a lot of yeast signatures also. Another 6 to 8 months time, we should be coming out with a big paper from the data what we have generated.
Okay, and Dr. Mohan Kumar, please.
Sir, are there any evidences of terrestrial becoming into ocean and vice versa, ocean into terrestrial?
Yes, Mohan, thank you for raising that. It's an interesting question. When I was talking about training to grow this, we did a vice versa experiment also. I took some bacteria, the fungal cultures which were isolated from the coastal areas, and we tried to train them to grow in the laboratory. What we did is, like, initially we grew them at 50, then we slowly increased to 100. And by the end of the cycle, we could say that out of the 20 cultures which we started, around 4 cultures are able to grow up to 400 bar of the pressure. So the same thing might be happening with these cultures when they are drifting down to the deeper parts of the water. That slowly they are, but then if you directly put them under pressure, like right from atmospheric pressure to 400 bar pressure, they do not survive. So you need to that is called as a gradual adaptation.
Okay, so how this metagenomics helped you in the identification, sir?
We
What?
have not done metagenomics for the data what I presented so far. This was all based on the microscopy. Metagenomic data is being generated from that expedition what I showed at the end of my presentation. That data is still not out. It should be soon out in form publication.
Thank you, sir. Thank you.
Dr. Patricia Willis, please.
Thank you very much, Professor Damare. It was a super interesting and exciting talk. And I'm curious, it's a technical question. Have you observed any growth differences in the isolates when you culture them in solid media versus liquid medium?
You're asking at the high hydrostatic pressure or the normal pressures?
Well, yeah, actually, my question was going towards the high hydrostatic culturing infrastructure.
Yeah. In the liquid media, we found a lot of differences. One of the slides where I showed the differences in the morphological, we could see a lot of swelling. That could be because of the pressure and the temperature. Solid, we couldn't see any differences as such. So maybe their agar, what we use, could be acting as protector. We don't know, because agar, most of the time we could see that it was getting turning into jelly at high hydrostatic pressure. It actually doesn't stay as agar at very high pressures. So the solid ones, we do not have much evidence, but liquid, yes, we saw a lot of morphological differences when the culture is grown at 1 bar and when it is grown at high hydrostatic pressure.
Oh, I see. So what would you expect for solid culturing conditions? I mean, you have no data, but what's your hypothesis?
We might get differences, but it will not be as much as in liquid, because what we did is we had done some experiments with the spore germination. We did a pore plate technique where we embedded the spores in the agar gel, and we cut those discs like how we do for the antibiotic susceptibility test. We cut those discs and we put them under pressure, and we could see that the germination of the spores was better as compared to the liquid. So same thing might happen for the mycelial growth also, but then we need to start with the mycelia. If we start with spores, we don't see that much of a difference. If we start with mycelia, we might see a good difference.
Thank you very much.
Thank you. Dr. Manjusha, please.
Yes, sir. It's very interesting and an exhaustive work, sir.
Madam, we can't hear you. Madam, can you please fix your sound? Okay now. Yeah, thank you.
So I was wondering, when you're incubating in the under high hydrostatic pressure, in what kind of a container are you placing the sample? As you said, the agar turns into gel in these conditions. So
Yeah, most of the experiments what we have done are with liquid media. We have not used so much of agar, but there is a special plastic which is gas permeable. We use that plastic for the gaseous exchange between the surrounding water. So the cylinder what I showed in my presentation, there we fill it with seawater, and then we suspend those plastic bags with the culture and the medium into this, and that is how we carry out the growth.
Okay. Thanks. That's very. Was that designed by your lab? The
culturing technique is what we have developed in our laboratory during my doctoral work.
Okay. Thank you so much.
Professor V.V. Sharma, please.
That's a wonderful presentation, Dr. Samir. I'm just wondering whether there is any possibility of this filamentous fungi becoming yeast forms via microcycle conjugation through a process of reductionism. Is it possible?
could be quite possible the way we have seen the microconidation and the different adaptations. But unfortunately, these what happens is when you expose them to the atmospheric pressure, they come back to. We need a cyclic pressure generator where they could be cultivated without any contact with the atmospheric pressure. Because once we open the pressure container, all the conditions come back again to the atmospheric pressure. So we cannot do a real continuous cultivation under pressure. That's one of the drawbacks with that. NIOT at present is developing something where we could do a continuous culturing, but they are also not so successful to keep it more than 10 to 12 days. So we need a fermenter kind of thing where we can grow and do the recycling of the nutrients. Then it will be possible to answer this question. With this, we have seen only up to microconidation. We have not seen anything beyond that.
Thank you.
Melisha, please.
Good evening, sir. It was really informative session. And my question is, did you ever found any chances to find mushrooms or rather macrofungus within the ocean?
We could isolate a couple of Basidiomycete, but then most of out of the cultures what we got, around 40 to 50% cultures we have lost during third or fourth subculturing. So there was one Basidiomycete which grew, but we couldn't take it to the fruiting stage.
Okay, sir. Okay. You can please name them? Any which
No,
you found?
we couldn't identify them because it couldn't survive. It just died after 4 subculturings.
Okay, okay,
If you are
okay, sir.
interested, you can just write a mail. I can send you the pictures of those.
Sure, sir. Sir, from where I can get your mail
It's
ID?
Samir at NIO, samir@nio.org.
Okay, okay, sir. Thank you. Thank you so much.
I just want to add one point about the Basidiomycetes, especially mushroom species present in the deep sea or in the marine systems. What we found, we have some data for the DNA signature of mushroom taxa in deep sea sediments. DNA signature doesn't mean mushrooms live there. All right. You can just Google and find out mushrooms taxa in deep sea sediments. DNA.
Yeah, I would like to add a plus. Mushroom is a fruiting body which is basically prepared out of the air. So in submerged condition, I don't know whether it will really fruit.
The possibility is very slim, but you just have a look at it. Yeah. Thank you, Dr. Devdatta. Thanks for your patience.
Thank you, sir. Wonderful talk, sir. Sir, I have a question about like how long do you incubate your samples? I mean, during the isolation process, how long do you incubate the media for the proper growth of the fungus?
Yeah, we have gone up to 90 days, but that is basically for the first incubation when we get the sediment from the deep sea and add nutrients to that. And that is where we use many a times 1:100 diluted nutrient and then leave it under pressure for 60 to 90 days. And subsequently, when we start growing them at atmospheric pressure, we incubate 15 to 30 days, depending on the visible growth which could be seen in both the agar as well as the liquid media.
Thank you, sir. Sir, as we are facing a lot of viability problem in marine fungi, so how do you preserve them for long term?
Yeah, I've started lyophilizing the cultures and keeping, and like last 10 years, my collection has grown to more than 1,000 of the fungi isolated from different extreme environments from the Indian Ocean. So lyophilizing is the best option. It's very difficult to maintain them, especially in a humid place like Goa, where you have a lot of contaminants coming in. So lyophilization is the best option. Go for that.
Sir, as of now, I have been reviving a lot of cultures from ampules. Still, we are facing a lot of revival problems from lyophilization also. So, like, can you suggest any other best method?
Are you making the ampules yourself? What is the media you are using there?
Sir, we are using we are on the media like properly grown cultures on PDA. We are adding skimmed milk for 15%, and then we are undergoing for lyophilization. So it was. I mean, as of now I'm in MTCC. We are reviving a lot of old cultures. So I have been facing a lot of like revival problems. To be frank.
Yeah, we use milk, skim milk, and DMSO sometimes.
Thank you.
May I add one point here? I mean, deep sea culturing can be very tricky, and I suggest Dr. Devadatta to go for DNA bank in MTCC for marine microbes. Thank you.
Thank you.
Dr. Rohit Sharma, final comments about this talk.
Yeah. Thank you, Dr. Shnath. Thank you, Dr. Samir. I think, what should I say? You have explained everything from conditions of the deep sea very clearly to the sampling tools, especially, that must have been very excited. And apart from that, I think you have come a full circle from the isolation up to the kind of complete story is full circle up to the roles. And most importantly, you clearly mentioned that the presence of the spore and the growth of the fungi in the deep sea is very clear. They are two different aspects and they have to be taken differently. So that's very nice. And at the last, I'll say that since it's a very high financial stake research, so we can understand the kind of accuracy needed for you to from the sampling up to the preservation of the organisms. So congratulations on your work. Yeah. Thank you so much.
Thank you, Rohit.
Thank you, Dr. Samir Damare, for enlightening us. It was a great presentation today, full of energy and passion, as I told you before. Thank you so much. Yeah.
Thank you.
Now I request Professor V.V. Sarma Garu to give his expert views or expert comments about the whole seminar. Professor Sarma Garu, are you there?
Yes. It was a wonderful day with very enlightening lectures from leading mycologists, marine mycologists from India. Starting, of course, I just started, but then in-depth analysis came from Professor Sridhar about the ecology of marine fungi, though he started as West Coast, but a lot of ecological aspects were covered by him, followed by Dr. Suryanarayan sir, of fantastic lecture about the endophytic fungi, different aspects of endophytic fungi, and then followed by Calaban, how they are bringing out those the online websites for different groups of fungi, which is very helpful, particularly for the beginners, because they have to search a lot. In those days, we used to visit the libraries like anything, get the data, note down in small card, like postcard size notes like that. Nowadays, you just click and then you get plenty of information. But then maintaining such websites is also important. And the Dr. Hyde group and Gareth Jones group, they are maintaining those online databases. And then followed by Varda, she has enlightened us about the thrastocytids and the different aspects of ecology, and also the biotechnological aspects of thrastocytids have been very well explained by her. And then, sorry, we have Dr. Patricia, who has explained about the Deepsea fungi and various biotechnology aspects, including the antimicrobial activity of the fungi from there. And then the culminating lecture is by, of course, in between I missed Dr. Narsim Thakur, who has explained about the marine fungal secondary metabolites and the research on marine secondary metabolites. And the last talk by Samir Damare, he has taken really into the deep sea world the advantage of those cruises and the devices, probes that they have. It is somewhat making me jealous that we don't have experiences things, but then they are doing wonderful work, and the class of information that we have got from him is also wonderful. And I thank Dr. Shanai for thinking about such a wonderful seminar, international seminar, and then making it possible by approaching different speakers and then arranging it cohesively in a single day, because normally this kind of lectures will take 2 days, but he could arrange it in one single day. I hope all the beneficiaries, means the young budding mycologists, marine mycologists, it will help them a lot in their future research pursuits. So it's a wonderful day. I thank all the participants and also all the speakers for their valuable sharing of information. Thank you.
Professor Salmagaru, thank you very much for your kind support. You were the actually support I was looking for, and you give that support to us actually. And thank you very much for making it happen today, and for your wonderful overview of the marine fungi, and for being with us. Probably you missed Dr. Manjusha's lecture too.
Oh, sorry. Yeah, that's what I was watching. So, marine yeast, yes, she has covered. And luckily, we have somebody in India, within India, who is working on marine yeast.
Yeah, and now I request any of the speakers if they want to share any comments about today's seminar. Anyone? Yes, Samir, please.
Yeah, it was a nice collection of the speakers, but then I would suggest, like, if we have next series of talks, something, try to get somebody from molecular biology background for a
feedback from.
Kapil Raj, don't interfere. Thank you.
Yeah, I was saying that now with all the people moving towards molecular biology, it would have been good a couple of speakers were there to talk about the NGS data and other things. So maybe for next, try to get one speaker in that.
Yeah, we try to get some foreign speakers, but we could get lucky with only two speakers, Dr. Calaban and Dr. Patricia.
Okay.
Thank you very much, Dr. Patricia and Dr. Calaban, for your kindness. And with this beginning, probably we'll probably climb the next height of marine fungi event. Actually, thank you, Dr. Samir, for that comment. Actually, we will definitely consider that, and we thank you again for your kind participation. Anyone else, please? Anyone from the Okay, Dr. Patricia, please.
Oh, I only want to thank the organizers for this excellent forum for discussion on marine mycology, and personally thank you for making it possible to meet one of several of my mycological heroes. But I always have read Rakukuk Kumar's and Damare's papers, so it was an honor getting to meet professors in this seminar. Thank you very much.
Yeah, thank you, Dr. Patricia. Thank you so much for spending your Sunday morning with us. Thank you so much. Okay, Professor Suri, please.
Can you hear me?
Yes, sir, we can.
So, it was a wonderful session, and we could see the diversity of work. So, marine fungi have attracted the attention of earlier the taxonomists. Now it is moving towards technological users. So, I suggest that all people who presented their talk, then there are others who are also working on different aspects of marine fungi, could think of the possibility of impressing on the government to have a consortium to look for novel metabolites, maybe a joint proposal, especially aiming at novel antibiotics to manage drug-resistant microorganisms. I mean, one could think on these things because each one has some speciality, and these specialties do not meet. But if we come together, that could be a good proposal, and that could possibly be we could end up with a couple of chemicals that would be useful in managing this AMR pathogens. So that's my view. Anyway, it was an excellent session. I thank the people who organized this. Thank you.
Any more speakers? No, I guess. I would like to thank all the speakers, for spending their valuable time and knowledge with us, and not only with the other speakers, with the budding mycologists of the world about marine fungi. And my special thanks goes to Dr. Rohit Sharma. Rohit, if you can switch on your video, please.
Yes, Dr. Shnoy. Thank you so much.
I mean, you have been wonderful spending time with us since morning 8:45, evening 6:45 p.m. And your comments, your summary of the talks have been very interesting and very helpful. And you have lessened the burden of my burden. Actually, thank you very much.
Thank you.
Great companion for me.
Thank
And we hope that we will continue this kind of association and
Yes,
in future also.
Yeah.
And if you have anything to say, you can please say.
Yeah, it was very enlightening for me. A learning experience, a whole lot of complete information on marine fungi. Since I have not focused on this area in my research work, so it was complete learning experience for me. That's all I would like to add. Yeah.
Yeah, I would like to thank Dr. Romana Mirde, who has been with us since morning. Thank you, Romana, if you're here, and also MycoAsia team, Professor Sridhar, editor-in-chief of MycoIndia, our editor-in-chief of MycoAsia, Dr. Naren Vijayavardhane, and the managing editor, Dr. Rajesh. I mean, the whole teams of MycoIndia, of course, Pondicherry University have been awesome. We hope this collaboration will continue, and I will now share the feedback form. Please fill the form in next 15 minutes. This is just to avoid the misuse of this feedback thing. Okay, thank you so much, and I hope everybody will have a great day, night, evening. Thank you so much. Bye bye.